All right, now we have a series of 3 trainee talks, and the first is Sushma Kalmodia from Children's Hospital, Los Angeles. Sushma.
So first of all, thanks to organizer and CIRM for this wonderful conference. Since we already heard about IRD a little bit, I will be talking about one of the IRD which we are working at CHLA. So this disease is mostly affect the kids around 3 to 15 years of the age. I will briefly talking about the retinal degeneration, pathology of the BEST disease, and I will tell about what is the treatment possibility and how we are trying to model this disease. So we know the eye is one of the important sensory organ in the human body which allow us to pursue the visual information, and retina is in the innermost layer in the eye. Any dysfunction in any of this retinal layer will be impair the visual function. So BEST1 is the gene which expressed in the RPE layer, which is the outermost layer in the retina. And the communication between RPE and photoreceptor is very important for the sight. The gene BEST1 is expressed in the RPE, and here we can see that one of the very important function of the RPE is to phagocyte the photoreceptor outer segment, and when the RPE is dysfunctional, photoreceptor outer segment is not phagocyted by the RPE, and there is accumulation of the lipofuscin. Since we know that there are more than 200 genes involved in the IRD, and one of the gene is the BEST1, most of this mutation in the BEST1 is missense, which cause frame shift. And this is very diverse disease genotypically and phenotypically. And here we see that in the OCT image, the whole integrity of the retinal layer is destroyed. And one of the characteristic feature of the BEST disease is the accumulation of the fluid in the beneath the RPE layer. There are a lot of complications of this disease as it's a macular disease, and macula is responsible for the central vision. We see that with this disease, central vision is lost. It's mostly affect the macula, so a macula is rich in the cone, and so also it's very difficult to see the color, and also it reduces the ability of a patient to pursue the colors and details. There are many stages of the disease, like choroidal neovascularization, where the new blood vessel form, geographical atrophy, where the cells start dying, and macular hole. In very advanced stage of the disease, macula is die, and the patient lose vision all of a sudden. The gene is expressed in the chromosome 11. It has 11 exons, and the variant which right now we are working at CHLA is on exon 7 and exon 3. So my rest of the talk will focus on the tyrosine to cysteine, which is at 284. This protein is involved in a chloride channel, which is activated by the calcium. So this is the subject we have at CHLA, and we will see that it's very characteristic feature of the BEST1, where is a bilateral fundus image changes, which accumulation of egg yolk-like appearance of lipofuscin, and in this particular variant, there is a large involvement of the macula. This variant has identified in Australia as well as in Mexico. So this is the recurrent missense variant. Since most of this variant bring a dominant negative effect, although we have one normal copy, but the mutant copy is always dominant. There are possibility to treat this disease with the gene augmentation, but that will not work for all of the dominant variants. So now it's time to solve the problem. So we think is the gene editing will help to solve this problem of mutation specifically in the BEST1? If yes, what will be the most clinically translational correction strategy? Recently, there has been some advancement has been made in treating the inherited disease by base editing, which was developed by David Liu group. There are two base editors. One is cytidine base editor, which converts C to T, and the other one is xanilin base editor, which converts A to G. There's two important components in the base editing. One is deaminase, which will deaminate the base, and the other one is nickase, which make a nick. So advantage of using base editing is it won't make a double strand break. So that's good because we don't want to make any double strand break while treating the patient. So this has used by David Liu group itself, and they show the DNMT1 gene, 48% editing using the cytidine base editor. At Palczewski group at UCI, they also use adenine base editor and edit the RP65 gene, which is responsible for the visual cycle in the retina, and shows 29% editing efficiency. So now we know that base editing could be a strategy for us to treat the BEST1 patient. We design our editing strategy. Since there are two base editor, so we use the negative strand where we have the C. Using the cytidine base editor, it will change to T, and ultimately on the coding strand, we have tyrosine. Before going further, we check the expression of the BEST1 in the RP cells to confirm what is the status of the BEST1 in both this variant, and we use a control line. It's to our surprise that in both of this variant, we see the full-length protein, but at the same time, we also see some other transcript variant, and that's also reported not for this variant, but some other variant in the literature. So now we know that that strategy can be for us to further try. The first step for us to generate the iPSC from the blood, so we collect the blood from this patient and create an isogenic control line and differentiate the iPSC to RP cells, and then ultimate goal is to deliver the base editor using the virus. So first we create the iPSC using our stem cell core facility SCHLA. We did quality control check, confirm the genotype, and then we use this clone for our further experiments. The first step is to generate the isogenic line where we use a guide RNA for that particular variant and the base editor. Since this base editor has a reporter, we are able to sort the cells, and then we check the editing efficiency, which is around 20 to 14%. That's vary between clone to clone. After having this efficiency in the bulk, we successfully able to get the single clones, multiple clones, and we use them further for the phenotypic experiment and other studies. So Dr. Daniel Clegg is here, and we use his protocol for this particular variant. We try other protocols also, but we have great success using this protocol. It's around 77-day long protocol. We have a little bit modification, but exactly we use the same factors which was published before. So then once we able to get the RP with this protocol, we have edit and a mutant line both, and to our surprise, we will see a big difference in terms of the morphology between a mutant and the edited line, and we will see that in a mutant line, there is a fluid domain formation, which is a characteristic of a Best disease. In the edited line, we will always get a very uniform polygonal shape and pigmented RP versus in a mutant line, that's not the case. Then we look for the trans-epithelial resistance, which is a characteristic of epithelial cells tight junction, and to both of the clones which we use for this particular experiment, the TER is very high, which is very significant compared to the unedited line, which signifies that this mutant is disturbing the RP monolayer, and the tight junction is not properly formed. We look for, and then we characterize the iPSCs for the BEST1 because that's the protein we are working. And in the mutated line, we have seen that mislocalization of the BEST1 versus in the edited line, its expression is very much localized to the basolateral site. We also quantify the BEST1 and ZO1. In both these cases, the mutant line is very low in terms of the expression of the both the proteins, ZO1 and the BEST1, but in a corrected line, we see the higher expression of these two proteins. While doing this RP differentiation for mutant line and the correct, we realize that there is a lot of cell death in the mutant line compared to the normal. We also quantify and see is this inducing the apoptosis, which is the case. In a mutant line, we will have around 13% of apoptosis. So our next plan, since now we confirm we have the RP, now we would like to deliver the BEST editor using the AAV because AAV is already in clinical trial and there has been great success for the Luxturna for RP65 gene. So we use AAV intine construct where these intines come together by trans-splicing, form full-length protein, and we use triple transfection method to produce the virus. Before going for the further editing, we first test the transduction efficiency of this virus construct, and we will see that in both the here. That this is able to transduce the RP cells. At the same time, we also produce the AAV which express the GFP. So now we did high throughput sequencing to see whether we are getting the RP editing by this intine. Since we don't have any way to see and sort the cells, and in the patient we are not going to sort the cells, so we need to see what is the editing efficiency in the RP monolayer. Since we remember that it's a heterozygous patient, so half of the population is already normal and half is the mutant. So here we can see that when we submit the sequence for the high throughput sequencing, it's a targeted sequencing. The 50% of allele is showing A instead of G, which is the case in the patient because the half of the allele is normal. When we use the virus, we see that the percentage of A is increased here, but at the same time we have seen some bystander editing also. And this is percentage of editing is increased when we increase the virus concentration, and that signifies that and the dose which we use here is clinically relevant, which is in the same range which our clinicians are using for the treatment of other IRD. Here we see the distribution of allele around the cleavage site, and in this untreated samples where just the cells, we'll see both the alleles is around 48%. But when we use, I'm showing data only for the highest concentration of the virus, we see that it's going down the mutant one. We also look for the indels, and in this case, there is no indel except the substitution, and that's what we are working with this. So our conclusion that this BEST1 variant induced the apoptosis, and we successfully able to differentiate multiple clones. We see that there is some improvement in the morphology, TER, and decrease the apoptosis. And also this is split intines able to edit the RP, which is around 10 to 12%. There is no off-target editing, and there is no significant indel formation in edited RP monolayer. Our future direction is to test this construct in the in vivo model, so then we can know what is the tolerability and safety of this split intine. Also, we are planning to identify the other sites, so then we can test our editing strategy and generalize this editing strategy, not just for this variant, but other variant of the BEST disease. And then we think that in future, if this works, we will try to potentially generalize this for not just the BEST disease, but other dominant IRDs. I would like to thank my mentor, Dr. David Kovanik, Dr. Aaron Nagel, Dr. Jennifer, and Dr. Lee. It's a great support from them, and we have funding from Knight Templar. Our core facility, Esteban, for always help in imaging. Narina is our stem cell facility. Andrew and Kayla, they work closely with on this project, and members of the Nagel lab, stem cell goal facility, and Dr. Kobernik lab, especially Jinjulin Pai. He shared his knowledge on cloning and other aspects. And thanks to CIRM.
Hi, I had a quick question. Where exactly are you doing this research? Is it at an academic institution or?
Yeah, so CHLA is Academic Institute, and we are working at CHLA.
How do you get your the patient-derived blood? How does it work when you're working at an academic institution and also working with like a hospital?
So the CHLA is a hospital, so it's there is already a study there where they are collecting blood from many of these IRD patients. So that's in the hospital, and we get the blood from the hospital and use our stem cell core facility to further get the iPSCs.
Thank you.
All right, we should move on. Let's thank Sushma again.
Thank you.